people have any questions they'd like to ask about Chapter 19 on RNA metabolism and transcription and processing before we get too far into the... Yes? I'll probably post a practice exam after this class. Other questions? The practice exams should be like the final stage of preparing. In other words, you should do the studying part first and then use the practice exam near the end of the studying. So it's not the beginning of your studies, but the end of it. Other questions or concerns? All right. Let's roll into protein synthesis then. So we talked a little bit on Friday about how RNA does seem to have some capacity for catalysis as well as information. But it's very clear that in life as it exists today, the great majority of enzymatic activities are contained in proteins or in polypeptides. And so in order to make those proteins, there has to be some process for taking this linear information of an RNA molecule that is in a nucleic acid and translating it from a nucleic acid into a polypeptide. And we're going to see that protein synthesis consists of a cycle of repeated steps and what they contribute. And in that cycle, RNA is on the stage playing a number of different roles. It's the messenger RNA, which contains a sequence information, and by itself is about as interesting as I don't know if anybody still has tape recorders or not, you know. I guess there's really nothing left that's a linear piece of information in daily life. There are ribosomal RNAs that play both a structural and a catalytic role in the ribosome. And then there are the transfer RNAs that have the job uniquely of matching up nucleic acids and adapting that somehow or other to an amino acid. Interestingly, in protein synthesis, there's a very large role for GTP. And we're going to see that GTP, in more than one place, is involved in generating a directionality to the reactions. Simply generating a linear polypeptide by itself is still not enough. As you remember, proteins have tertiary structure, if not quaternary structure, so there has to be folding. In many cases, there is processing that goes on for proteins, and some proteins are exported either into the membranes or out of the cell altogether. The diagram on this particular slide is an antibiotic, and this is one of the few but important exceptions to polypeptides being made through translation. The polypeptide antibiotics in general are made by enzyme complexes where there's a separate enzyme for each amino acid in the antibiotic. And looking at those enzyme complexes makes it compellingly clear why we need the kind of machinery we have for protein translation. We simply can't afford to build machinery that does each thing one step at a time. So I'd like to remind you then a bit about the triplet code and the implications it has for how translation happens. We've got 20 amino acids. And one of the insights that Francis Crick gave to this whole question was realizing that if there are 20 amino acids and there are 4 bases in nucleic acids, then there has to be at least a triplet code. It might have been a quadruplet code, but it had to be at least a triplet code. And the reasoning goes like this. Let's suppose we have 4 bases, and you can have any of those 4 bases in each of 2 positions. So there's 4 in the first base, any of 4 in the first, any of 4 in the second. That's 16 possible doublet codons. Not enough for 20. By having 3 positions, 16 times 4, so we have 64 combinations, more than enough for what we need, and we'll see how these extra or redundant codons get used. Now, there are a number of features of the actual genetic code that we use that were not logically necessary. They are just simply properties of the code. It is a sequential code. In other words, you read it, you read a chunk of it, and then you read another chunk of it, and then you read another chunk of it. It's not an overlapping code. So you read 3, and then you go to the next 3. It could have been you read 3, and then you go down 2 and read some more, but that isn't how it happened. It is a degenerate code, and by degenerate we mean that if I give you a protein sequence, you don't know what the RNA sequence is. Since there are 64 codons and all of them get used, when you see a leucine in a protein, you don't know what the codon was that gave you that. It could be any of several codons. And so the genetic code is degenerate. And oftentimes when we say that, we also add unambiguous. That if I give you an RNA, it will only make one protein. Another feature of the genetic code is what's called frames. So if we look at this particular sequence here and block it off into 3s, if we start at the A, then that gives us threonine for the first codon, isoleucine, a serine, arginine, and so on. Suppose we had started making the triplets just one down at that C and took these 3 nucleotides, then we would have had proline, serine, arginine, glutamine. That's quite different. If we had started in the 3rd position, we'd have had histidine, leucine, glutamate, serine. Again, quite different. So where you start reading the triplets is called the frame, the reading frame. This is one reading frame. This is a second reading frame starting there. And this is a 3rd reading frame. And so that adds another dimension to the question of how do we match nucleotides to amino acids, because there has to be some way to select the frame in which to read these triplets. So this is one of the presentations of the standard genetic code. Just to sort of refresh you on how this is read, the first position in the codon is over here, the second up there, and then for each of those there's 4 different things. And so you can see that if your codon is C, U, anything else, you mean leucine. If your codon is U, U, if it could be phenylalanine for these 2 or leucine. So we have, this is an example of its redundancy or its degeneracy, if you want. The other thing you can pick up from here is that they're not scattered all around. So phenylalanine is near leucine, is near isoleucine and valine. So you've got these very hydrophobic ones in codons that are related to each other. You have over here codons that are, or here perhaps I should say, you have codons that are polar or charged and so on. So you don't have a completely random distribution of the codons. It could have been the case that for phenylalanine you used UUU, CCC, AAA, CGA. It would have been possible, but it would have been much less robust. That by having the codons for a given amino acid clustered and having the codons that are for amino acids similar to each other also clustered, a change in one codon may end up not changing the protein or at least changing it in a conservative way. And this magic is accomplished through transfer RNAs. So somehow or other, the transfer RNAs are going to have to become charged with an amino acid. We call that process tRNA amino acylation. You need to make a covalently attached amino acyl group. And so the tRNA is going to be what it is that can read codons. It has one end that's called an anti-codon loop. And at the other end is going to be where the amino acyl addition is going to happen, where we put on the amino acid covalently. Some tRNAs are called isoacceptor tRNAs. So they are tRNAs for one particular amino acid, but they might be able to read more than one codon. We just saw that there are 6 codons for leucine. And so it's possible to have 2 tRNAs read all 6 of those codons between the 2 of them. And to give you a feeling for how common that is, bacteria have 30 to 40 tRNA genes, so they actually have fewer than 60. You know some of those tRNAs are reading more than 1 codon. Higher organisms may have as many as 150 tRNAs, and the exact function of those extra tRNAs is a separate question. tRNAs are fairly small RNA molecules, 60 to 95 nucleotides. It's quite common for tRNAs to have one or another type of covalent modification. We talked about that a little bit. So here's a pseudouridine over here, and other features like that. It's also very common for there to be secondary structure. So this is a bit of double helix in here. Here's some double helix in there, and there, and there. That whole structure as a whole is going to fold up into a tertiary structure. There are 5 common features that are found in all all tRNAs. You have an anticodon loop, because that's going to be what can read it. You have the acceptor stem, that's going to carry the amino acid. And the D loop and the T5C loop are common features, not identical in each one, but with similarities. And then a variable loop that's of different sizes in different tRNAs, anywhere from 3 to 21 nucleotides long. This is what it looks like if you kind of flatten it out with an iron and you want to see the codon relationships. I think the next picture is going to, yes, show you more of a sense of it as a 3-dimensional thing. If you look at the transfer RNA structure like this, you can appreciate that it's a compact thing. It's slender, so that it's sort of like a boomerang in some respects. It's slenderer than it is long. And because of its slender shape, you could imagine packing several of these side by side and yet having the anticodon end still be able to fit in there. tRNA tertiary structure has a lot of non-standard base pairing. So we mentioned before in the RNA part that although Watson-Crick base pairs are the common sort of standard form, there are a variety of others that can occur, and those are much more common in things like tRNAs that are sort of structural RNAs. The bases, for the most part, are out of contact with water. There's a lot of stacking interactions among the bases. And it puts the anticodon end a long way away from the amino acyl group. So it's 75 angstroms from one to the other. It separates them by quite a distance. As you can imagine, this isn't something that can happen spontaneously. There's an enzyme that can catalyze this reaction, and it's called an amino acyl tRNA synthetase, or AARS. And the amino acyl tRNA synthetase has the job of joining the correct amino acid to the correct tRNA. And it's going to take a couple of steps. Here's an amino acid that we want to charge onto a tRNA. We'll spend 2 high-energy bonds to create an amino acyl adenylate here. So you have a high-energy bond in there from that reaction, and it has enough energy in it to be transferred to a transfer RNA, and so this is now covalently attached. Here's the covalent attachment between the amino acid and the tRNA. So this is why it's an amino acyl tRNA. Since what comes off is AMP, it costs us 2 high-energy bonds to do this. And you can see here the amino acyl group coming off of the 3' hydroxyl of the terminal adenine of the transfer RNA. Now what's interesting, and it's sort of one of those things of, golly, gee, is that there is one amino acyl tRNA synthetase for each amino acid. That one AARS will do all the tRNAs for all the codons for that amino acid. And so if there might be 6 codons, one AARS manages to charge all the tRNAs, whether there's 2 or 3 or however many, for that amino acid. And this is a picture of the contacts between the tRNA and the AARS, in this case for glutamine. And you can see that the two components make a lot of contacts. It's almost like it's the teeth sliding between two combs or something. And in the AARS, there are some domains that can read the codon to determine, which one we're looking at here. They're not reading it with RNA. They're reading it by various kinds of contacts from the side chains. And then a separate domain catalyzes the addition of amino acid at the CCA end, or the aminoacyl end. So you can imagine here one end doing the determining about which codon is there, and the other end doing the catalysis. I've heard it described as the ARS sort of sniffs the tRNA all over. Some of these enzymes are even able to proofread. As an example, the isoleucine tRNA synthetase has a way to keep out amino acids that are bigger than isoleucine so they can't get charged in. But if it's something smaller than isoleucine happens to be charged on, it will hydrolyze that at a proofreading site. So an additional layer of accuracy. So in reading the genetic code, the trick here is we're going to have 3 bases in the tRNA matching to 3 bases of the messenger RNA, but it's a little bit anti-intuitive, so be patient with me. Nucleic acids always bind anti-parallel, so we know that the contact between the tRNA and the messenger RNA is going to be anti-parallel. But the convention among biochemists and molecular biologists is to write all sequences 5' to 3'. And so if your codon is UUC, its anticodon is GAA. And that's a little easier to see down here where there's actually a picture of it. So if you read the messenger RNA 5' to 3' UUC, its anticodon coming back from the other direction is GAA. Okay. The third position of the codon, which is here, which makes it the first position of the anticodon, has sometimes able to wobble. That is, that one position may be able to read more than one codon. And inosine is often found in tRNAs in what's called the wobble position. You remember back when we were looking at the code overall that the third position was often the one in which we got synonymous codons, and that we can sometimes find tRNAs that can read 3 different codons that are differing in this wobble position. This is just showing you some more examples of messenger RNA codons and their anticodons. This is showing you how the inosine manages to read A and C and U in the messenger RNA, yet the one tRNA reads all of them. Okay, so now we have a tRNA. The tRNA has a specificity to it because the anticodon means one and only one thing, and at the other end it's got a covalently attached amino acid. How do we line them up so we can get them to make polypeptides? Well, that's going to be the job of the ribosome. And the ribosome can be thought of as a molecular factory whose job is to match up the codons and the tRNAs and then to catalyze the making of a new bond. And in this process, both the RNA and the protein are important to get that to happen. We know that it takes a lot of investment to make this work because 80% of the cellular RNA is the RNA in the ribosomes, this kind of structural enzymatic RNA that isn't itself informational. It doesn't have any information in it. It's just the machinery to make the information usable and visible. This is one of the subunits of a ribosome. This is from a bacterium. Bacteria and eukaryotes have slightly different sizes of ribosomes. Eukaryotic ones are generally 80S, and the prokaryotic ones are generally 70S. You notice I say generally because I did my graduate thesis on algae, and they're kind of in the in-between ground. But whether you're eukaryotic or prokaryotic, there are two major subunits to the ribosome. The large subunit would be either 50 in prokaryotes or 60S, and the small one would be 30 or 40S. The large subunit takes several RNAs to construct it. The small subunit has just a single RNA in it. And again, these units are Svedbergs. Svedbergs don't add up. You could say I have a 60S Svedberg subunit and a 40S Svedberg subunit, and it adds up to an 80 Svedberg ribosome, because Svedbergs don't add. So don't let it bother you too much. This is showing you just the RNA component of the ribosome, so you can see what it looks like without the proteins kind of tweaking it and giving it conformation. So the RNA molecule inside the ribosome subunits acts as kind of the reinforcing bar in concrete or something. It's going to provide structure to that thing with a lot more information than a rebar. It's going to provide binding sites for all the different subunit proteins. And when we look at the small subunit, there's some flexibility there, so it's able to wiggle to accomplish its job. This is a picture of a ribosome, and you can see here how some of the ribosomal proteins are at the surface, and some of them are somewhat deeper. Some of the ribosomal proteins have a lot of secondary structure. Some of them are just kind of wiggly spaghetti interacting with the RNA. The large subunit is less flexible, and it has a face that's designed to interact with the small subunit. And we're going to see why that would be in a second. So if it takes a small subunit and a large subunit to make a functional ribosome, those two subunits have to come together. The place they come together is the interface there. And as you might imagine, that interface is conserved because you need to make the thing hold together. The interface is also the place where the messenger RNA is going to be held or moved along, and the transfer RNA is going to be bound during the translation process. So you're really creating the location for all this to happen by bringing the two subunits together. And here you can see an intact ribosome. And in this case, they've bound onto it 3 transfer RNAs. So this is a fully loaded ribosome. And the ribosome has 3 kinds of tRNA binding sites. These names will make more sense in a little bit. An aminoacyl site, a peptidyl site, and an exit site. So the small subunit, or the 30S subunit, is going to be the one that checks the match between the anticodon and the messenger RNA to make that happen. And the large subunit is going to be the one that recognizes that things are set up right now and catalyzes the formation of a new bond. Now you'll remember that I said, given the fact that we have a triplet code here and that there are frames to read, there has to be some way to pick out where to start. And so one of the important parts of protein translation is initiation. That just as there's a time and a place to initiate DNA synthesis or to initiate RNA synthesis, protein also has important things happening. In prokaryotes, there is a sequence in the RNA called a Shine-Dalgarno sequence. And this sequence in the prokaryotes is complementary to part of the ribosomal RNA. So that acts as kind of a little Velcro spot to help the RNA identify the right spot to grab onto a messenger RNA. And the Shine-Dalgarno sequence is about 10 bases from the first AUG. AUG is methionine. And it turns out that every protein, when it starts to be translated, starts with a methionine. Some of them get cut off later on, but every protein starts with a methionine. So that's a codon that has kind of 2 jobs. One job is methionine, wherever it is. In the middle of a protein, at the end of the protein, it means methionine. but it also means start here in terms of translation. Eukaryotes do not have Scheindel-Garneau sequences. They, for the most part, are going to read from the first AUG, although there's some fog about that. But even though this is coding for a methionine, there is a special tRNA that reads that first AUG. So there's an initiator tRNA that reads methionine, and then there's a plain old tRNA that reads methionine everywhere else. And prokaryotes make it just that little bit more interesting by having a 4-mil methionine as their first methionine. So this is a picture here. You can see the methionine there. And they've added a little 4-mil group here. And that the initiator tRNA is charged with methionine and then has this little modification added onto it. So prokaryotic proteins all have a 4-mil methionine in their initial state. So we've got to get together messenger RNA, tRNA, and 2 big pieces of ribosome. So we need to get stuff together. So let's start by looking at a small subunit here. The small subunit is going to bind a FMET tRNA, the initiator tRNA, and messenger RNA. So we need to get to a complex that looks something like this. Small subunit here, messenger RNA bound. Here's the FMET tRNA. But that isn't going to happen all by itself. Initiation factor 3 will open up an empty ribosome to provide a small subunit for all this to happen. And initiation factor 1 is going to sit and occupy the A site so it doesn't get taken up too early. Initiation factor 2 has GTP bound to it. And when it delivers the tRNA FMET, the initiator tRNA, to that AUG, when it successfully delivers that, it hydrolyzes the GTP into GDP and then comes off so it can go around and catalyze another round of initiation. And so this small subunit with its tRNA and messenger RNA are a friendly place now for a large subunit, sort of looking like an elephant here, to come in and sit down and complete the ribosome. And so now we have a fully assembled ribosome. It's positioned, the messenger RNA, so that the AUG is aligned with the P site, the peptidyl site. And it has room now for an A site. It has the A site empty, so it can accept an adenoacyl tRNA into that site for the next round. So there's initiation. So there's one place where GTP comes into it. In eukaryotes, it's a little more complicated, and we're not going to try to piece it apart into all of the specifics. There are in the order of 9 initiation factors in eukaryotes. We're still going to use an initiator methionine tRNA. That is, there still is a specific tRNA just for starting proteins with. And we're still going to definitely need messenger RNA, of course. Some of the other things that are around in kind of the eukaryotic zoo are cap binding protein. You may remember we talked about the methyl G cap on messenger RNAs. Well, there is a protein that binds to that cap, and that protein, when it's bound to that cap, is acting as kind of a tag saying this is an intact messenger RNA that has a proper cap on it. There's helicases. You know, we've talked about how RNA can develop secondary structure. You need to be able to open that out. There's a poly-A binding protein. We talked about adding extra A's at the end of messenger RNAs. Those are recognized by poly-A binding protein. And interestingly enough, even though the poly-A tail is at the far end of the messenger RNA, down at the 3' end, it interacts with some of the initiation factors. And so a fully competent, assembled eukaryotic messenger RNA is a circle. It turns out that that's one way that the cell has quality control, because if you insist that there's a poly-A binding protein with a poly-A at initiation, you know that there's some place to go. If the only thing that was your control was the cap binding protein, your message could be only half long, and you'd waste all the work of making half a protein and have nowhere to go. If you insist on it having a poly binding protein, then you know you have the full-length messenger RNA. The small subunit then in the eukaryotes consumes ATP as it scans along a messenger RNA. So it's going to be looking at that messenger RNA, and that process depends on ATP hydrolysis to keep the message moving through it. And it's looking for an AUG that's in a context something like this. So there's the AUG that will be the methionine, and there's some sequence around it that is what makes it the right first AUG to do it. EIF2, to get into sort of the way these names get named, the E here is for eukaryotic, IF for initiation factor. So EIF2 is eukaryotic initiation factor 2. It delivers the tRNA to the P site. We saw that had to happen for prokaryotic initiation. There's again a round of GTP hydrolysis. And then the large 60S subunit can come in and complete the ribosome. So that's initiation. We have a completely assembled ribosome with a messenger RNA that's correctly positioned with an initiator tRNA in the P, the peptidyl site. From now on, what we're going to do is elongate. We've got the first one sitting in the right spot. We need to now add amino acids for every 3 nucleotides or for each codon until we get to the end. And that's elongation. And so in every cycle, there will be an amino acyl tRNA that enters at the A site, the amino acyl site. It doesn't get in there all by itself. There is an elongation factor that assists with that. And it's a GTP binding protein called EFTU in the prokaryotes. And this is a picture of a tRNA here and EFTU bound to it. EFTU is a completely ecumenical protein. It recognizes all tRNAs. So it doesn't care what the codon is, the anticodon is. It doesn't care what amino acids on the other end. It's mostly looking at the T5C loop and the acceptor stem, the double-stranded stuff in there. And it provides kind of a handle to that amino acyl tRNA to deliver it into the A site. And so the tRNA is going to have some measure of affinity for what the codon is at the A site or the amino acyl site or the acceptor site. And that affinity is based on how good a complement there is between the codon and the anticodon. These diagrams are kind of interesting because they give you a sense of how tight it is. This is actually drawn pretty much to scale here to show you here's the A codon and here's the P codon. This is the E tRNA in the exit site getting ready to leave the ribosome. And the ribosome actually has to bend the mRNA a little bit to make enough room for that. You can see here a little distortion in the backbone in order to accommodate all of the tRNA, even though they are kind of skinny. And they check for the exactness of that fit. And the diagram here is showing you some of the, at the level of the molecule, how that checking goes on. So. Elongation is said to have something called kinetic proofreading. So if there is a correct fit between the anticodon and the codon, the correctness of that fit is transmitted to EFTU through the ribosome and so forth, and EFTU will hydrolyze the GTP and then go away. And so we're talking then about this tRNA fitting at the A site. If there isn't a good fit, if it's close but not right, that whole complex, the amino acyl tRNA and EFTU, can leave without that GTP hydrolysis happening. And so it can go and try another time on a different ribosome and see if it can get a good fit on that ribosome or on that message. Protein synthesis is pretty good. Given what it's trying to accomplish, it's amazingly good. We talked about the extremely high fidelity of DNA replication, of the considerable fidelity of RNA transcription. Protein synthesis has an error rate of about 1 in 10 to the 4th. So if you have if you have a protein that has 1,000 residues in it, one of them is probably wrong. But given the fact that proteins don't live forever, and given the fact that proteins don't make more proteins, these errors don't accumulate. The problem with having errors in DNA is that all the cells that go on from ever after would have that mistake. Error rates like this can be tolerated in protein synthesis precisely because protein is the end of the line and the protein molecules don't live forever. You could think of it as good enough accuracy. This is just a diagram showing you the kinetic proofreading. You have here your loaded ribosome that's ready to receive a tRNA. If the one that comes in is the incorrect aminoacyl tRNA, it can occupy it for a while, but it's not going to have a very good affinity for it because it doesn't have a good match there, and the whole complex will come off and be available to use somewhere else. After doing however many trials of that, when the correct one does come in, that will be signaled to the EFTU and have hydrolysis of the GTP, so this goes off now, and you now have a ribosome set up with a growing chain at the P site or peptidyl site and a freshly delivered correct tRNA at the A site. Those of you with sharp minds are going to think, well, this has GDP on it now, and this has GTP, and yes, there are other proteins that catalyze exchanging the GTP for the GDP. If that looks anything like G proteins to you, there's probably a reason for that. One of the things this means is that to have that level of accuracy of 1 in 10 to the 4th, we have to invest a GTP per amino acid. So we invested 2 high-energy bonds to charge the tRNA through ATP. Here we're using a GTP to have it come and sit down in the correct place. So the next step is going to be an activity on the enzyme called peptidyl transferase. And what we have diagrammed here is the ribosome in 2 states. So this is the first of the 2 states, and this is the second of the 2 states. The actual catalysis of the peptidyl transferase is the catalytic activity of the large subunit, of the 50S subunit in us. The energy for it comes from what was invested earlier by the charging enzyme, by the ARRS. So the amino acyl bond in the tRNA has the energy that's needed to transfer that bond over to the next thing. And let's take a little detail here. Here's the P site, the peptidyl site, with a tRNA in it that has the growing peptide chain. We don't know how long it is. Here's the A site that has the newly delivered tRNA. And the new bond is going to be made between the amino group of the new tRNA that's in the A site and the carbonyl carbon of the peptidyl group. So this whole chain is going to be transferred from here to there. And so what this means is that proteins grow from the amino terminus to their carboxy terminus. You're always adding on at the carboxy terminus when we do protein synthesis. So just as nucleic acids have a favored direction of polymerization, so do proteins. Nifty fact, on the large subunit, the actual catalytic residues that accomplish peptidyl transferase activity are RNA parts of the ribosome. So this is one of the most, well, this is, we could say, the most important RNA catalyst we know, possibly a survival of an earlier time. The catalysis here is accomplished by 2 things that we talked about earlier when we talked about enzyme mechanisms. proximity and orientation, so that they're holding it close to each other in the right spot, and they're holding them, pointing the right direction for the next thing to happen. So it's a good example of proximity and orientation accomplishing catalysis. So here we are. We have now an empty P site and an empty tRNA in it, and we have an A site that's got the growing peptide chain in it. We don't want to stay there for very long. This is just a transient fact. The next step to change that is called translocation. And translocation is when that peptidyl tRNA that's got the growing chain slides from the A site into the P site of the ribosome. The empty tRNA, the one that used to have the peptide chain on it, also slides down one into the E site. And the messenger RNA slides down one codon. So the ribosome can ratchet it through a codon at a time. And this also takes sort of the assistance of a protein factor called elongation factor G. And this is the picture of EFG. It kind of looks like EFTU, except that you've got protein where the RNA would be. So it's possible that what it's doing is bumping that growing peptide chain down. But it has GTP bound to it. And when it hydrolyzes the GTP, it leaves the ribosome. And then we have restored the situation where we have the growing peptide chain in the P site and an empty place in the A site to do it all over again. And so we can call this the elongation cycle, that we have to do certain things multiple times. So if we start with a ribosome like this that has a growing peptide chain in the P site, EFTU delivers the correct tRNA into the A site, hydrolyzing a GTP. The reaction called transpeptidation or peptidyl transferase moves the growing chain from the P site tRNA, attaches it to that new amino acyl tRNA, leaving the empty one. The next reaction is translocation. Again, it costs us GTP, and after it sort of has sat in that site, it hydrolyzes, comes off, and we're back where we began. And we can do this again and again and again. This is quite processive. One of the proteins I used to study quite a bit was 1 million molecular weight as a single peptide chain. And so the ribosomes that translated it had to keep going for a very long time. You notice that there's a GTP used at the delivery step and a GTP used for translocation. The transpeptidation does not require any additional input. Its energy is all inherent to the tRNA because we spent the 2 high energy bonds making it. So in terms of the processes that are going on the ribosome, it's 2 GTPs per peptide bond made. So this can go on for a very long time. And if you have a protein of 1 million molecular weight, it can go on for 10,000 times. At some point, it has to come to an end. And just as we've seen for the other macromolecular processes, that termination is distinct from the other 2 phases. There's something that has to play a role for protein synthesis to come to an end as well. And we call this termination, and it's accomplished by release factors. And release factors can read in the A site. So this is a ribosome that looks like it's all ready for something else to come in and keep going. But at this point in the messenger RNA, it has a codon that says stop. There are 3 codons that say stop. And so there's no tRNA for them. But there are release factors for them. And so if there is a stop codon there, a release factor can come in and bind at that spot, and it will trigger the ribosome to attempt to transfer the peptidyl chain over, but since there's no tRNA there to transfer to, the peptide chain comes off. And so after triggering this transfer, you have the free polypeptide chain, an empty tRNA there, and a release factor in the stop position. This complex by itself is not going to do anything more. We have to somehow or other get it apart from all the pieces. And so we have a third release factor, RF3 GTP. So a GTP is bound to the active form of the RF3, and it catalyzes the release of RF1 or 2 and hydrolyzes GTP in the process, as so often. And then that allows, that leaves behind a ribosome that looks like this. But you may remember that the initiation factors had the job of opening that up to get an empty small subunit for the next round. These release factors are, again, an example of sort of molecular mimicry, if you like. They kind of look like an anticodon end of a tRNA. So they can bind at that spot, even though they're peptides and not tRNAs. So, okay, maybe that's a good place to break for the day. People want to call that it?